AS Level Biology 9700, Paper 2, Variant 2, February March 2026, question number 1. And question number 1 is complete table 1.1 to show which features are found in typical animal cells and which features are found in typical plant cells. Put a tick in the box if the feature is found in the cell and a cross if the feature is not found in the cell. Put a tick or a cross in every box. APS ribosomes in the cytoplasm. They are present in both animal cells and plant cells because both of them are the eukaryotic cells, so they are present. Cell wall composed of peptidoglycan. Cell wall is absent in animal cells, but the cell wall which is present in plant cells, it is composed of cellulose, not peptidoglycan. So the peptidoglycan cell wall is found in bacteria, which is a prokaryote. Third one is the large permanent vacuole. Many small vacuoles are present in animal cells which are not permanent, but in plant cells, a large single permanent vacuole is present. So, question number 1, part B, feature 1.1. Figure 1.1 is a diagram representing cell in phloem tissues in a mature leaf. Some of the features of the cells are shown. You can see phloem sieve tube element, cell A is there. You can see mitochondria. You can see plasmodesmata. And in figure 1.2 is a diagram representing part of the cell surface membrane of cell A, in which they have shown the proton pump and co-transporter protein. Now question is, describe and explain how the structure of cell A is related to its function in transporting sucrose into the phloem sieve tube elements. In your answer, refer only to the structures that are visible in figure 1.1 or figure 1.2 or both. As I have told you that in the figure 1.1, we can easily see uh, the number 1, the most importantly, the mitochondria. So the mitochondria in figure 1.1. So produce ATP via aerobic respiration to provide the energy required for active transport. Produce ATP to provide energy. Uses ATP to actively pump hydrogen ions out of the cell, and you can see the proton pumps are there in figure 1.2, and co-transporter proteins are also present in 1.2. Before going to figure 1.2, I think I would like to discuss the plasmodesmata, which are visible in figure 1.1. So, you know, the plasmodesmata provides a direct cytoplasmic channel allowing the accumulated sucrose to readily diffuse from cell A into the phloem cell sieve tube element. So I would like to write this over there. That is they provide provide a direct cytoplasmic channel which allow the accumulated sucrose to diffuse easily. And in figure 1.2, you can see the proton pumps which use ATP to actively pump hydrogen ions out of the cell A into the cell wall space. Establishing a high electrochemical or proton gradient. Proton gradient outside the cell. And co-transporter protein it will allow hydrogen ions to diffuse back. To diffuse back into the cell A, down their concentration gradient. So all these four features were important over here. Now, the next question is, explain how sucrose that enters the flowing sieve tube elements from cell A is transported to a sink. So you know that the transport of sucrose to a sink takes place by the mass flow mechanism. So first of all, the water potential when the entry of sucrose into the phloem sieve tube elements lowers its solute water potential, then water enters the sieve tube elements from the adjacent xylem or surrounding uh, cells down a water potential gradient by osmosis. So the entry of the water increases the hydrostatic pressure inside the sieve tube elements at the source end. Then At the sink, sucrose is actively uploaded and used or stored, which increases the water potential in the sieve tube, causing water to leave and lowering the hydrostatic pressure at the sink end. So sucrose moves dissolved in sap from an area of high hydrostatic pressure to an area of low hydrostatic pressure down a hydrostatic pressure gradient by mass flow. So in this way, the uh, sucrose enters the phloem sieve tube elements of cell A to a sink. So first of all, I would want to write this all the entry, the entry of sucrose into the phloem sieve tube into the phloem sieve tube element lowers its water tension. Water enters the sieve tube from adjacent xylem or cells down a water potential by gradient by osmosis. The entry of water increases the hydrostatic pressure inside the sieve tube, causing water to leave and lowering the hydrostatic pressure at the sink end. So sucrose moves dissolved in sap from an from an area of high hydrostatic pressure to an area of low hydrostatic pressure down a hydrostatic pressure given by hydrostatic pressure gradient by mass flow. So students, it was all about question number 1. And please like, share and subscribe my channel to watch more videos on this paper. Thanks for watching.